right. Good afternoon, everyone. Thank you for joining us this afternoon. As you all know, we are here to celebrate Caroline Rodriguez, who will be defending her master's thesis this afternoon and sharing all the research she's been doing as a graduate student here at Moss Landing Marine Labs. So before we begin, I'd like to introduce myself and share a little bit about what it's been like to have Caroline as a master's student here in the lab and a little bit about her journey to her project. So for those of you that don't know me, my name is Dr. Cheryl Logan. I'm a professor in the Department of Marine Science at California State University, Monterey Bay, and I've had the pleasure of serving as Caroline's primary research advisor for her thesis. Joining me today in this introduction is Dr. Amanda Kahn, who has also served as one of her co-advisors here at Moss Landing Marine Labs, as well as Dr. Tom Oliver, who's at NOAA Pacific Islands Fisheries Science Center. Before we go any further, I also have a few housekeeping announcements. So we have a live audience here at Moss Landing Marine Labs, as well as an online Zoom audience. I am here in person. Caroline is going to be presenting via Zoom from Washington, DC. I would like to ask all of our online participants to please keep their audio muted and their videos off, and please do not attempt to share your screen while she is presenting. There will be a time to ask questions after her presentation, and I'd ask the online participants to please use the raise hand feature, which is located under the reactions button on your Zoom window. And then we will also have questions from the live audience here. So let's go ahead and begin. So for many of you that know Caroline, you know she is a true water girl. She spends lots of time underwater. Growing up, she was a competitive swimmer and a coach for a kids swimming club. And when she got to college, she also served as the president of the swim club at Duke University. Beyond swimming as a kid, as she got older, she also found a love for scuba diving. Long before grad school, she was certified as a scuba diver. And then during her journey as a graduate student, she became an AAUS scientific diver as well as a NOAA diver. And through her love of the water, this also spurred a love for environmental science and marine science. Caroline graduated from Duke University in 2013 with a double degree, BS in environmental science, as well as a BA in earth and ocean science, with an important Spanish minor that she has used extensively. Her very first introduction to working with NOAA was as an REU Sea Grant, an REU student for Maryland Sea Grant. And she also had her first foray into international volunteer and research work in Quito, Ecuador, where she was a research assistant for Acción Ecológica. Following graduation, she went on to another international volunteer experience where she served in the Peace Corps in Nicaragua between 2015 and 2017. And there she was acting as an environmental educator. And it was during 2017 when Caroline first reached out to me for potential research opportunities in my lab at CSU Monterey Bay. And the very next year, she joined us in 2018 into the Moss Landing Marine Labs Master's in Science in Marine Science program. She was awarded a very prestigious fellowship upon entering the program. It's called the NOAA CCME, or Center for Coastal Marine Ecosystems Fellowship, which funded the majority of her research while she was here. And as part of this program, this enabled her to have a 12-week internship at a NOAA facility. And specifically, she worked at the Pacific Islands Fisheries Science Center in Hawaii to learn what it's like to work for NOAA. And as part of this program, students are encouraged to learn the ropes of what it's like to be at NOAA, with the goal of some of these students then going on to have careers at NOAA. And you'll hear later that currently Caroline is serving as a Knaus Sea Grant Fellow through NOAA in Washington, DC. So she's really taken advantage of what this program has had to offer her. So Caroline's primary research has been focused on temperature effects on coral demography in Hawaii. But prior to her jumping into that project, she also served as a research assistant with me in the Galapagos in 2019, where we were trying to identify particularly thermally tolerant coral individuals in the Eastern Tropical Pacific, which is a location with a really long history of bleaching and oceanographic features that are similar to what we expect with climate change in the future. And fortunately, this was prior to the pandemic when we were still able to do lots of in-person research. And Caroline and I got to know each other very well in the field. We were doing a lot of diving together, collecting corals, placing them into this portable thermal stress system that you can see back here. And we measured coral thermal tolerance in 2 different species across 7 different islands within the Galapagos. Caroline became very familiar with coral reef ecosystems through this project and learned how to collect corals, identify corals in the field, and learn about coral thermal physiology. Upon getting back from the Galapagos, she immediately began her NERTO internship in Hawaii, the first summer of graduate school. And for this project, she was working with Dr. Tom Oliver, who would later become one of her co-advisors for her master's work. And this summer, she was learning how to use photogrammetry data in order to look at coral demography in Hawaii coral populations. And she led a paper that came out of this internship that you can see here in 2021, measuring coral vital rates using structure for motion photogrammetry at fixed sites. She has been an excellent scholar here at Moss Landing Marine Labs. Through her research, she's been able to present at 2 large international scientific meetings, including the International Coral Reef Symposium this past summer, as well as the Ocean Sciences meeting in 2020. She's also been the recipient of numerous scholarships and awards. So as I mentioned, she is currently a NOAA Canals Marine Policy Fellow in Washington, DC. She started this fellowship in February 2022 and will be there through February 23. This is a highly competitive fellowship that Caroline was selected for. There were only 4 fellows from the entire state of California. She's also been a recipient of the MLML WAVE Scholarship 2 years in a row, the CSU Biotech Program Travel Grant in 2020, the Women Divers Hall of Fame Graduate Marine Conservation Scholarship in 2019, and as I mentioned before, she's been a fellow within the NOAA Center for Coastal and Marine Ecosystems Graduate Research Fellowship Program between 2018 and 2021. Caroline has also been nominated twice as the CSUMB Undergraduate Mentor of the Year Award. And this goes in part to serving as a mentor to 3 excellent undergraduate researchers at CSUMB, Melissa Vizard, who's currently a master's student at the Bren School for the Environment at UC Santa Barbara, Kaiku Kaholoa, who is a first-year PhD student at Stanford University, and Lita Dawson, who is finishing up her degree this year at CSUMB. You can also see in the background that there's a lot of images of corals and delineations, which you'll hear more about with Caroline's research in a few minutes. But it took, a whole army of not only these undergraduates, but other staff members at NOAA to help produce all of the data and the imagery that Caroline used for her research. Caroline has also been just an amazingly wonderful lab member in the Logan Lab at CSUMB. She is very compassionate and kind. She's always giving other folks really constructive advice on their own research projects. She helped us to develop a Logan Lab Code of Conduct, and she also participated in the OpenScapes program, which is a program that our lab participated in to increase our ability to conduct open and reproducible science. She's also been a fantastic Moss Landing community member. She's always up for a good time, whether she's bowling, backpacking, spelunking, or just out on the boat. You never know what you're going to get, but she has some really amazing photos of her time here at Moss Landing. And not only that, she's also been a wonderful leader. She's been the CSUMB liaison for the Moss Landing student body, a co-facilitator for the Future Leaders in Marine Sciences program, and she served on the leadership committee for the Society of Women in Marine Sciences. But Caroline moves around a lot, and if you're ever trying to pin her down for a meeting or just a time to hang out, you have to invite her for ice cream. Caroline absolutely loves ice cream. So if you want to just enjoy a little bit of time with her, invite her out, bring her some ice cream, and she will be overjoyed. So Caroline, keep shooting high. You are amazing. It's been so wonderful to have you in the lab. We're all going to miss you, but it's so exciting to see what you're doing in DC And for those of you out there here in the audience live and on Zoom, I encourage you to follow Caroline on Twitter. She has been tracking her story from CSUMB to DC for her Canals Fellowship through this hashtag here. And you can learn about all her adventures in DC as she continues this amazing fellowship. So with that, Caroline, I'm going to go ahead and hand it over to you. And thank you all again for being here today.
All right, Cheryl, can everyone hear me, hopefully? Cheryl, thank you so much for that amazing introduction. I really appreciate it. I'm really excited to be here with everybody today to talk about my master's research, measuring the impact of thermal stress on coral reef resilience in Hawaii using large area imagery. And I'm excited to show you guys what I've been working on for the last couple of years. All right, Theo, unless there's any problem, I'm gonna move forward. Good to go? Okay, awesome. All right, so coral reefs. Well, first off, just to start off, you'll notice at the bottom, there's going to be a legend that'll help keep track of where we are throughout the presentation. All right, now coral reefs, the most important part. So corals are invertebrate animals, they're not plants, and coral polyps are tiny soft body organisms that are related to jellyfish. And Theo, are we okay? Okay, I'm assuming so. All right, and for colonial hard corals, where thousands of individual polyps are cemented together by calcium carbonate skeletons that they secrete. So why do I care about coral reefs? The first time I went diving was in Bali, and everyone after the dive was talking about all the amazing fish. And I just responded by saying, well, the fish were kind of in the way, but the corals were particularly beautiful. And I immediately wanted to learn more about corals and coral reefs. In addition to corals' intrinsic value, their beauty and cultural importance, they're also extremely valuable to everyone sitting in this audience today. So coral reefs provide ecosystem services worth $11 trillion.
Recording in progress.
Worth $11 trillion annually. Corals protect coastlines, sustain fisheries, and they're important for food and livelihoods. They also generate tourism and create jobs across the tropics. However, there are several anthropogenic threats in red. There's overfishing that depletes fish populations that control competitive algae from outcompeting coral diseases which can wipe out populations, runoff that promotes algae growth, and most importantly, climate change stressors, including ocean acidification and rising temperatures. While corals are threatened by several threats, ocean warming is the most widespread and immediate threat to coral reefs globally. And the frequency and intensity of these events is rapidly approaching unsustainable levels. And looking at this figure on the right-hand side, showing over time, looking at the red axis, that the cumulative number of severe and total temperature stress events since 1980 is increasing. And looking at this blue axis, it's also showing that there's a depletion of locations over time that remain free from any disturbances. So there's going to be, it's projected there will be more and more frequent thermal anomalies over time, and fewer refuges from global warming. And thermal anomalies can lead to coral bleaching, which can negatively impact corals at the individual and population level. Warmer temperatures can result in a stress response where the symbiotic relationship between a coral host and its photosynthetic algae breaks down and the algae is expelled, turning the coral white. So you can see here that the healthy coral, when exposed to thermal stress, expulses its algae, and this can lead to death. However, corals can tolerate short periods of bleaching or even recurrent bleaching events, but they do need time to recover and regain their photosynthetic algae following a bleaching event. And bleaching can negatively impact corals in several different ways by increasing partial and total mortality. And then it can also negatively impact both the reproductive output of corals and the growth rate, so corals' abilities to increase in size. And bleaching recovery is difficult to predict, partially because there's numerous reasons or factors that affect recovery. And recovery can be dependent on taxa, which is species or type of coral, and morphology, so the physical attributes of corals, where certain taxa might be susceptible to bleaching, as well as certain morphologies like branched and corymbose corals, which are both susceptible to bleaching, while other taxa and morphology like massive and encrusting corals are resistant. And corals can be divided into winners and losers based on certain traits, and these include both taxa and morphology. So this figure on the right-hand side is showing a PCA, which shows clusters of samples based on their similarity. And similar groups will cluster together and then are also in the same color. So looking at stress-tolerant corals, these are domed with large corallites, and they have slow growth but high fecundity. An example is massive porites. And there's also competitive species that are large, branching, encrusting species that grow quickly, but bleaching can cause high mortality. And this includes branching Poeciloptera and encrusting Montipora. And I want to take a quick shout out, take a break to also introduce these illustrations, which were created by Mina Satari at Moss Landing. I will be using these illustrations throughout the presentation for mounding porites, branching Poeciloptera, and encrusting Montipora. Recovery time can also vary drastically between reefs. It can take less than 7 years or anywhere from 10 to 12 years or more. And in this photo here, you see in the first quadrant that corals are bleached. In quadrant B, there's fleshy algae growing, then a full seaweed bloom, and then finally in the last quadrant, it's actually a recovered reef. And this time to recovery depends on various factors, and it's like environmental conditions and thermal history. And these conditions can be site-specific, which means that you can have two reefs that are right next to each other that might have different recovery trajectories. And given the fact that bleaching events are increasing in frequency and severity, it's important to understand how thermal history affects coral populations. And while we know how that bleaching leads to increased mortality, stable or decreased growth rates, and then can negatively impact recruitment, it's unclear what the population level impacts of current or severe events. Which brings us to the first objective of this project, which was to explore the impact of temperature stress on coral genera with distinct morphologies. And changes in coral communities are generally studied by tracking coral percent cover, but this doesn't always elucidate the factors that are driving the community dynamics of corals. So looking at this figure here, showing the percent coral cover change, where anything above the line means it's increasing in coral cover, anything below it's declining. And so you can see that while generally speaking, these columns are showing that overall there's not that much change in coral percent cover. However, when you look at the demographic processes, you see that there's actually highly active demographic dynamics at play. So for example, a lot of corals are experiencing significant growth, like you can see here in this color, and shrinkage in this color across the different time points. So demographic approaches, unlike coral cover, can reveal the drivers of population structure and recovery. And these are done with demographic metrics called vital statistics. And these include growth, so a coral growing from one time point to the next, mortality, where it was alive and then the next year, unfortunately, passed away. And then reproduction, or what we'll refer to as recruitment throughout this presentation. And these vital rate statistics or vital rates show how fast the vital statistics are changing in a population. And when combined, vital rates can be used in population modeling, which are useful in projecting recovery trajectories. And most importantly for this talk, population models can be used to estimate the population growth rate, or lambda. So you're going to hear this term a lot, and that's just the change in the population over a certain time period. And Hawaii is an ideal system to investigate bleaching, susceptibility, and resilience because of its thermal history. So the Hawaiian archipelago, as you can see here, is comprised of the main Hawaiian Islands and the northwestern Hawaiian Islands, or the Papahanaumokuakea Marine National Monument. 16 sites were selected because they fall along a gradient of thermal stress event frequency and thermal stress and severity. So the frequency is showing the number of thermal stress events in a 10-year period. Severity is, we use this metric called degree heating weeks, or DHW. And degree heating weeks show how much heat stress has accumulated in an area over the past 3 months by adding up any temperature exceeding bleaching threshold during that time period. Basically, it's quantifying the coral's exposure to temperatures above a bleaching threshold. All right, so I'm going to walk everyone through this relatively crazy plot. So as a reminder, the degree heating weeks are showing our accumulated heat stress. Anything higher than 4 degree heating weeks is usually indicative of minor or moderate bleaching, while higher than 8 degree heating weeks usually it means that severe bleaching is likely. So looking at all of the colored lines here, every single peak you're seeing is a thermal stress event. But as you can see, looking across these 16 sites, there's both different severities where some peaks are higher than the other, and there's also different frequencies, so different numbers of thermal stress events compared to different sites. So zoning in just the main Hawaiian Islands, you can kind of see that there's these thermal stress events in 2014 to some degree, 2015, and again in 2019. Versus when you look at the Northwestern Hawaiian Islands, there's a much more active thermal history, to say there's much more frequent thermal stress events. And finally, the last thing about this plot is that the vertical gray bars are showing our sampling periods where coral monitoring was conducted. So the Hawaiian archipelago serves as a natural experiment to assess the population dynamics of dominant coral genera with distinct morphologies. All right, so now I'm going to move into my research questions. So the first question was how do coral population growth rates differ between 3 genera? So massive porites we expect would be the most resistant to thermal stress, similar to other massive corals, and the population growth rate will be increasing. And branching Poecilopora species will be the most susceptible to thermal anomalies, and the high mortality following bleaching events will outweigh their traditionally fast growth and lead to decreasing population. Finally, encrusting Montipora species grow quickly and are capable of recolonizing an area following disturbance, but temperature stress can cause high mortality. So these populations will initially decline following a bleaching event and then stabilize in the long term. My second research question looked at which vital rates most influence the population growth rate. As a reminder, vital rates are growth, survival, and recruitment. And we expect that life history differences between taxa will affect which vital rates drive lambda. For porites, recruitment is likely to have the largest effect because changes to their high fecundity could reduce the population growth, any changes to their fecundity. Both Poecilopora and Montipora will be most impacted by growth because their fast growth rate is necessary to maintain the population despite their susceptibility to bleaching. And then my third question looked at whether the frequency and or severity of thermal stress events correlated to changes in the coral population growth. And it's likely that we'll see decreased survival and recruitment as a result of recurrent or frequent thermal events that will lead to a decreased population growth. And then corals exposed to more severe events will also suffer due to decreased survival due to the rapid onset of a severe event. So overall, the two of these variables will lead to decreased population growth rate, or lambda. All right, now we're going to move on into the methods section. So first off, this is the overall workflow for our methods. And the first step involved monitoring corals over time, and we did this using something called structure for motion photogrammetry. We used this to build 3D models of reefs and monitor them over time. The next step was collecting the demographic data, which were then used in population modeling to project the population growth rate. So the first step. So collecting coral demographic data can be very time consuming. As you can see in this photo, there's divers underwater collecting individual colony level data while scuba diving. And so we needed a novel method that goes beyond this traditional method of just monitoring coral percent cover. So structure for motion allows us to do that. In this photo, you see 2 divers underwater that are taking thousands of overlapping photos of the reef bottom. And in about 15 minutes, you can collect these thousands of images, which are then stitched together in a computer program to build 3D coral models that are also just beautiful to look at. And by revisiting sites, the 3D models can be used to observe colony level changes over time. So after we created the 3D models for each monitoring year, we aligned them in this software by dragging them on top of each other. And the software is not currently capable of measuring 3D change, so the aligned models were exported into 2D into what's called orthomosaics. And these orthomosaics were used for demographic annotation. And annotating the entire reef plot, this 10 by 10 meter plot, is extremely time consuming. So we did random subsampling. And even with this random subsampling, some of these plots took more than 20 hours to annotate. So the way this works is that we census the population in each time point. This is an example of looking at coral or reef in 2016, again in 2019, and then comparing the 2 years. So we compare the patches over time based on spatial overlap. So this example where you can see that from 2016 to 2019, the red annotation in 2019 is larger, which indicates a growth event. Looking over here, this annotation in 2016 of a little coral no longer appears in 2019, and this is a mortality event. And then finally over here, you see that the small red annotation from 2019 is where a coral appeared where it was barren space before, and this indicates a recruitment event. So for each live patch, we collected species, genus, planar area, max diameter, perimeter, as well as a unique ID number, which was used to link patches across time points. And this was used to establish growth or shrinkage, mortality, and recruitment. In total, we annotated 1,025 Montipora colonies, 264 Poslopper colonies, and 2,790 Porites colonies. And these were censused from 2013 to 2019 for a total of 51 orthomosaics annotated. And I want to take a quick second to provide a huge shout out to the annotators, both at NOAA, Pacific Island Fisheries Science Center, as well as the 3 wonderful CSUMB undergraduate researchers. And this Herculean annotation effort would not have been possible without all of them. So using these vital rate data, we're able to build a population model called an integral projection model, or IPM. And an IPM uses how an individual state, the age or size, influences vital rates to project changes in a population. And as a reminder, that output is lambda, which is the population growth rate. And while it's logical to divide the life cycle of some organisms, like plants or insects, into discrete classes, coral demographic processes are more strongly linked to size than age. So for example, a lion at a certain age reaches sexual maturity, and at this age is able to create little baby lion cubs. But for corals, the size of the coral is a more important factor in determining when a coral will be reproductive. And this is an example of, yes, when they'll be reproductive. And it is difficult to apply IPMs to corals due to their life history complexities. So especially reproduction, which is shown in this photo. And the other challenge is the insufficient data. But luckily for us, the structure from motion data provides us with the demographic data we need, which allows us to quantify the processes that are most likely to increase resilience to thermal stress, and then explore the impacts of environmental factors, including ocean warming, on reef population dynamics. So the first step of building the IPM is modeling vital rate functions. And first is growth. And growth for corals is very complicated because corals can experience partial mortality, where part of the coral dies. They can also experience fission, where lots or large coral piece breaks off into lots of little pieces. And then fusion, which is the reverse, where these little pieces fuse together. So for this study, growth was defined as the probability of each colony undergoing growth and shrinkage. This was modeled as a linear function. Survival is also relatively challenging. So corals can experience 2 types of mortality: partial mortality, and as seen in this GIF on the left side, total whole colony mortality. So for this study, survival was defined as a probability that a coral would not undergo whole colony mortality from one year to the next. And because this is bimodal data, either die or you live, it's modeled as a logistic regression. And while this study is one of the largest coral demographic studies, it does involve many different sampling periods, which complicated our IPM building and necessitated annualizing each vital rate. And to save time, I'm not going to go into this process except to say that we annualized growth and survival by dividing by the time interval between censusing. Finally, reproduction recruitment is also challenging. So first off, it's very difficult to observe a coral reproducing, unlike lions, where you can walk up and see the baby. As you can see in this photo, corals will release thousands of little babies. And corals also undergo sexual and asexual reproduction. We ended up modeling recruitment in 4 different ways. And again, for the sake of time, we won't get into that. But if anyone has any questions, I'm happy to talk about that afterwards. And so the recruitment parameter was estimated as the number of recruits per the amount of adult coral area. And IPMs were built for each region and genus morphology for a total of 6 IPMs. All right, for our results section. So I'm actually going to provide the results and conclusion for the first question, then move on to the second and then the third. As a reminder of our first question, whether if the population growth rates will differ between the 3 genera. So to walk you through this, any lambda value, which is the population growth rate, is on the y-axis. Anything above 1 shows that the population is increasing. Anything below means it's declining. And these are the error bars represent 95 confidence intervals, and the difference in lambda was determined by non-overlapping confidence intervals. And lambda values were similar for, population growth rates were different for each taxa in the Northwestern Hawaiian Islands over here. But looking over at the main Hawaiian Islands on the left side, lambda values were similar for both Passalopera and Porites. But in general, there's very low population growth rates because most of these regional models projected lambda values less than 1 or declining. And for example, in the Northwestern Hawaiian Islands, looking at this Porites lambda value of 0.79, this is a 20.9% annual decline in the population. And looking over in the main Hawaiian Islands at Passalopera and Porites, which had lambda values of around 0.86, this means that populations were declining by 14% annually. All right, so IPM contour plots. So these plots are used to represent the summed investigated demographic parameters and are used to observe the probability of growth and survival, as well as recruitment from the first time point to the next time point across all size classes. So we'll walk through this as well. The x-axis is showing the time in the first, the size, in the first time point. Y is size in the next time point. This diagonal band right here represents survival and growth. And the black diagonal line represents corals that are in stasis or not changing in size. The horizontal band at the bottom represents coral recruitment. Anything above this line is showing corals are growing. Anything below, it means they're shrinking in size. And in general, these warmer colors right here are indicating the increased probability of size transitions from one year to the next. All right, so for this one, rather than focusing on the individual plots, we're going to talk about some overall trends between the Northwestern Hawaiian Islands on the top, the main Hawaiian Islands on the bottom, and each column for each genus. So in general, the colors are a little bit lighter because when they're lighter, it means a lower probability. So when you do focus in on these warmer colors, you can see up here that there's a higher probability of survival for these larger corals up here at the top. But you'll also notice that these warmer colors are falling below the stasis line, which means that there's also a high probability of shrinkage, particularly for Montipora corals in the Northwestern Hawaiian Islands. So what this means is that corals are basically breaking up to avoid total mortality and still survive. In the Northwestern Hawaiian Islands over here, we had limited transition data, so there's no relationship between the size in the first time point and the size in the second time point. And this is just because in the Northwestern Hawaiian Islands, there's just not a lot of Passalopera corals. This limits our ability to draw conclusions. Finally, looking at the bottom, which is where recruitment is, it's showing that recruitment stems from the largest corals, as we expected. And focusing in on recruitment, you see that with our recruitment calculations, Montipora had the highest recruitment in both regions, versus Porites had the lowest recruitment in both regions. So what does this mean? So first off, in both regions, populations are declining anywhere between 4.6 to 21% annually. The exception is Pasalapra and the Northwestern Hawaiian Islands, if you remember, had exhibited a positive population growth. But due to the insufficient sample size, it's difficult to say whether or not this is an accurate representation. And we expected that massive Porites would experience a increasing population growth rate. However, we found that it is declining, and this is because, and it's possible that the limited recruitment could be driving this declining population growth rate. We expected branching Pasalapra to be susceptible, and we found that it indeed was. It had declining population growth. And we expected that encrusting Montipora would have a stable population growth rate. And it's possible that high recruitment, which is we were incorrect about, and it's possible that the high recruitment is not enough to offset the higher probability of shrinkage that Montipora are experiencing. And this could be a case where Montipora are allocating their physiological resources towards recruitment at the expense of growth. Also, overall, larger colonies had a high probability of survival, which is expected, but they also had this higher probability of shrinkage. And so this does fit in with the observed widespread partial mortality that we saw across the orthomosaics, especially for Porites and Montipora. And so this could be an example where it's actually biologically advantageous for larger coral colonies to fragment into smaller pieces to avoid total mortality. Moving on to our second question, which is looking at which vital rates most influence the population growth rate. So to answer this question, we did use the same methods as before, but with one additional step, and we added what's called an elasticity analysis. And these are used to determine the contribution of the IPM components, survival, growth, and recruitment, to the population growth. So elasticity analyses allow us to determine which vital rate had the greatest impact on the population growth rate, and therefore had the most impact on population recovery. So looking at these plots as a reminder for these ones, these warmer colors in this area are showing which vital rates within the IPM have the greatest impact on lambda, which is the population growth rate. And as a reminder, this region is showing our survival and growth portion of the IPM. Down here is showing our recruitment. And what you can see across all of these regions is that medium to large corals surviving, but also maintaining their size as they stick around this black line, has the most impact on lambda. So what this means is that losing medium or large colonies would have a large impact on the population growth. And similar to what we saw with the IPM contour plots, the elasticity analysis in the Northwestern Hawaiian Islands didn't show or was inconclusive. And this is, again, likely due to the fact that there's just very limited Pasalapra data. And if recruitment impacted the population growth rate, you would see dark red at the bottom. And so the only exception across all of the regions and taxa is that Montipora on the main Hawaiian Islands. And what this is saying is that recruitment is an important factor that affects lambda for this morphology in region. And it's also possible to separate the IPM kernel into the growth, survival, and recruitment components of the IPM. And this allows us to determine the relative contributions of each factor separately. And so looking at this, what you'll notice is that growth and survival contributed over anywhere from 95 to 99% to the lambda, whereas recruitment only contributed anywhere from 0.001 or less to less than 5%. And what this means is that growth and survival had the largest impact on lambda for both regions and across all genera. So we expected that life history differences between taxa would affect which vital rates drive the population growth, but we found that for all that larger corals surviving and maintaining their size, or not shrinking, not undergoing partial mortality, influences all other demographic processes. And the idea that survival of medium-large colonies drives population dynamics, supports this current understanding that survival and reproductive output increase with colony size. And we also found that recruitment had a minimal impact or effect on lambda. And so this could be that corals are surviving and maintaining their size despite thermal stress. And so they don't need that boost of recruits to maintain the population growth. And so what's important then is that, recruitment may not be needed to maintain the population size. And this has important implications for management, including that management or conservation efforts should try to focus on factors that affect survivorship of these medium to medium large corals. And this could include things like enhancing species diversity or, at a very small scale, nutritional enhancement. So feeding the corals just to ensure that those medium corals are surviving and able to grow. All right, for our third question. So the past two questions looked at these aggregate regional models between the Northwesterns and the main Hawaiian Islands. For this, our third question, we're going to look at IPMs for each site, interval, and genus. And this will help us determine if the frequency or severity of thermal stress events correlates to changes in lambda. So as I mentioned, IPMs were built for each site, interval, genus, and we used NOAA Coral Reef Watch data to estimate thermal stress conditions. And this photo down here is an example of one of the outputs of the NOAA Coral Reef Watch. This is showing sea surface temperature anomalies. And to determine if there was a relationship between the frequency or severity of thermal stress events, we estimated thermal conditions at each site. So for severity, we calculated the mean of all maximum thermal stress events, or showing how severe those events are. We also calculated frequency as the number of thermal stress events per 10 years, so how many events are occurring. And to evaluate the relationship between the frequency and severity of thermal stress and lambda, we correlated the output, which is lambda value, to each site's thermal stress signature. And we did this by using linear regression models to look at whether frequency or severity separately impacted lambda, multiple linear regression models to look at the combination of those two variables, and then chose models based on Bayesian Information Criteria, or BIC. Awesome. All right, so this is focusing in on the main Hawaiian Islands first. And you can see that from 2013 to 2019, anywhere that lambda is higher than 1, again, that means that the population's growing. Anything less than, it's declining. And these vertical salmon-colored lines are showing bleaching events that occurred in the region, and small horizontal lines are showing the sampling period because those differed, and the small vertical line is an error estimate. And finally, this blue line is the locally weighted polynomial trend. So for all three genera, Montipora, Placellapra, and Porites, there's some general trends you can see. So following the repeat bleaching events in 2014 and 2015, you can see that lambda values are lower in 2015. And you especially see this down here for Porites. And you start to see these lambda values increase in 2016 for Montipora. It takes all the way up to like 2017 for Porites. And this could be delayed recovery due to these very low lambda values following the repeat bleaching. What's interesting is that the lambda values start to drop again moving into 2018, 2019, especially for Montipora and Placellapra. And unfortunately, due to the pandemic, there was no sampling done in 2020, but it's considering that there was this bleaching event in 2019, it's concerning to see that lambda values were already starting to decline moving into this bleaching event. Now looking at Papahanaumokuakea Marine National Monument, we see similar trends to the main Hawaiian Islands, where lambda is less than 1 in the 2014 to 17 repeat bleaching time period. And for Montipora, you see up here that there's, during the 2016 to 19 period, there is a recovery where lambda values are higher, versus Porites down here, they're still exhibiting declining population growth rates during both bleaching years and recovery years. So now looking at the thermal stress modeling, and this we're starting off with Porites. So these individual plots display the relationship between lambda and individual predictor variables while controlling for the presence of the other predictor variable, And this therefore reflects the statistically independent effect of either severity and frequency. And so we anticipated that increasing frequency and severity of thermal anomalies would lead to declining populations. And that is exactly what we see here, that those combined variables negatively impact parietes. In contrast, when we applied the same model structure to Montipora, it resulted in a linear regression fit. So this means that we looked at severity separately and frequency separately. And in addition, it resulted in a fit with almost no explanatory power, which is to say there's no relationship between lambda and severity or frequency of thermal stress events. So the increasing frequency and severity of thermal anomalies did negatively impact parietes, but as I mentioned before, it didn't impact the encrusting Montipora. And it's possible that the ability of encrusting corals to quickly recolonize areas following disturbance events allowed them to overcome high mortality and maintain their population growth rate. But it's also possible that we can't explain this based on morphology or taxa, and that and other studies have actually found that coral family was explained more variation in bleaching response than morphology or physiological traits. And what's concerning is that if massive parietes, which are considered stress tolerant, are negatively impacted by frequent and severe thermal anomalies, it begs the question of what will the results for susceptible taxa like branching Poeciloptera be? And unfortunately, due to just limited Poeciloptera data, we weren't able to do this modeling with Poeciloptera. But it would be very helpful for future studies to see how these susceptible taxa fare. And in general, for our study, we looked at the climatological impact, or the impact of climatological thermal stress. But it would be interesting to look at acute stress, and this could be done by more frequent sampling. As I mentioned before, some of our sites were sampled up to 3 years apart. And so honing in on maybe 1 or 2 sites and repeatedly sampling them rather than trying to work across the entire archipelago like we did. In addition, it would be interesting to model additional environmental variables to get a more robust analysis of thermal stress. And this could include many other variables looking at thermal stress, other metrics that measure thermal stress, as well as things like water quality parameters that can have an impact on bleaching. So overall, for this study, we had a lot of accomplishments. As Cheryl mentioned during the introduction, we developed this process to estimate vital rates for individual coral colonies from orthomosaics. And this was published as a NOAA technical memo in 2021. And this project in general uses emerging technology, structure for motion photogrammetry, to understand the ecological crisis. And our study also represents one of the few applications of IPMs for demographically complex organisms like corals. And this is the largest coral IPM study to date, from our knowledge, in terms of spatial and temporal coverage, which is really big, a really big deal. And we also overcame a lot of modeling hurdles in this project. One of those was looking at those, building those aggregate regional models for each taxa and region, which was something that we hadn't seen been done before. And we also overcame issues with, we built models that had different time points or different amount of time between sampling periods. And both of these things were, spent lots and lots of time doing the actual modeling. I'm happy to talk about more of that if anybody has any questions, but they're definitely 2 big achievements from this study. So why do we care in general? So all 3 coral genera, which had different morphologies and life history strategies, had negative population growth rates. And this is really concerning for genetic diversity in Hawaii. And it's also concerning for coral cover in Hawaii, that these populations might be declining over time. It's interesting that population dynamics were primarily driven by coral growth and survival. And what's cool is that these can be targeted for future restoration and adaptation projects. However, the problem is that preserving large corals has a larger impact than something like artificial recruitment, because recruitment played a small role in maintaining populations. But it's much harder to control factors that impact coral survival, like temperature, and doing things like artificial recruitment are much easier. And so while recruitment, trying to boost up recruitment is still important, it's important for management to understand that growth and survival of these medium to medium large colonies is very important to the population. And then finally, the increasing severity and frequency of thermal anomalies from climate change is causing population level decline of what is considered to be a stress tolerant morphology. Our massive parietes are considered stress tolerant. So again, what does this mean for susceptible taxa like branching Poeciloptera? So I would like to provide some acknowledgements, first off, to my funding sources. I want to thank the NOAA Center for Coastal and Marine Ecosystems Fellowship and Corey, who I saw is online. The CCME program funded both my graduate education as well as the research for this project. I also want to thank Moss Landing for the WAVE scholarships that I received to help fund my education as well. The CSU PERB scholarship, this enabled me to travel to Florida to get hands-on field experience and learn about coral restoration. And then finally, the Women Divers Hall of Fame scholarship, which again gave me the chance to travel to the Galapagos for a once in a lifetime opportunity to learn about coral thermal tolerance across the Galapagos archipelago. And I also want to thank my data sources, specifically NOAA Pacific Island Fisheries Science Center, Ecosystem Science Division, for providing both the computing power and imagery from 2019. And a huge thanks to the Sandin and Smith labs at Scripps, as well as the 100 Island Challenge for providing imagery for the 2013 to 2018 reef models. So I want to thank the greatest thesis committee that ever existed. To start off, Dr. Cheryl Logan has been an absolute constant support throughout my entire time at Moss Landing. No matter what obstacles I came across, she was there to support me and wants me to succeed as a scientist. And in addition to, again, inviting me to go to the Galapagos, which was absolutely amazing and just the coolest thing I think I'll ever do in my entire life. She also welcomed me into her home. And so while I was in the Galapagos, I spent a fair amount of time living in the apartment next to Cheryl with her kids and her husband. And I really appreciate that time spent with them. Dr. Thomas Oliver, who is my NOAA mentor, is truly the most supportive mentor that wants to see me succeed as a scientist and has really worked to make me a better scientist through hours of coding sessions, but also by welcoming me into the NOAA community in Hawaii, and as well welcoming me into his home. And this is a photo of Charlie, the Hawaiian boar that Tom and his family fostered for a short period of time. And I was given the chance to even to hang out with Charlie. But again, being part of Tom's family and building that community in Hawaii really meant the world to me. Amanda, I don't have any embarrassing photos of you, but I want to thank Dr. Amanda Kahn. She, in her very first year at Moss Landing, agreed to be my Moss Landing faculty member, and her exuberance for science really is motivating. And I also want to give a shout out for the cat therapy sessions she's provided in letting me meet her cats to keep me going throughout some of the tough times of grad school. And then finally, I want to thank Dr. Thomas Connolly. Tom really brought the modeling expertise to this project. So I have never done modeling before this project. And his advice for writing about modeling and talking about modeling were absolutely critical. And there's also a lot of people I want to thank. So first off, I want to thank Dr. Joshua Maiden and Devin Wolstein, who are coding and modeling whizzes. They were absolutely critical for all of the IPM modeling. My annotation team, so NOAA Structure for Motion team, and then as well the three undergraduate researchers, Kai, Melissa, and Lita, again, for putting in hundreds of hours annotating these plots. Couldn't have done it without you. I want to thank the entire Logan Lab, especially the graduate students, and give a shout out to Holly Dorr and Melissa Noggle Everyone in the lab has been absolutely amazing, but Holly and Melissa have become some of my closest friends, and I really cherish their friendship. I also want to thank the Moss Landing Invertebrate Ecology Lab for supporting me throughout my entire time at Moss Landing. And the Moss Landing community, everyone, like Moss Landing is just awesome. All the students are amazing. Special shout out to Sophie Bernstein, who is part of the reason that I'm here today, for keeping me on track. I also want to thank the front office. I spent most of my time at Moss Landing working at the front desk, and Kathleen in particular is the most supportive boss in the world. And the shop guys and IT, just thanks for everything you do. And then I also want to thank my current job, NOAA Office of International Affairs. You guys have taught me so much in such a short period of time. And finally, last but absolutely not least, my friends and family. You know who you are. You've all been there for me throughout this entire experience, and can't thank you enough. And with that, I'll take any questions.
Maybe we're gonna have a participants window. If you see someone raising their hand in here, just call on them and I will unmute them.
Okay, great. Thank you so much, Caroline, for that wonderful presentation. We are now at a point where we can welcome questions either from the live audience here or those of you that are joining us via Zoom. At this point too, if you want to ask a question, you can, if you're called upon, you can go ahead and unmute and show your video as well. Okay. All right, so we have our first question from Sophie Bernstein. Sophie, if you want to go ahead and unmute.
Hi, Caroline. Great job. I had a question about the modeling. I know this was a lot of effort in R, and I was wondering if the script that you wrote is something that could be applied to other data sets. So for example, if similar data exists from other coral hotspots, if you can basically just clean the data and run it through your model and get not similar outputs, but get similar results. Because I'm wondering if this basically could just be taken at a much larger scale than what you've developed in Hawaii.
Yeah, definitely. Thanks for the question. Again, I think one of the big achievements from this project was figuring out how to apply IPMs to corals in a different way that's been done in the past. And so I think that, yes, this can definitely be applied to other corals throughout the globe. And the other really big benefit of our modeling effort is collecting the growth and survival data. Demographic data is, can be challenging, but really the hardest part is recruitment. And what I didn't get into here was we went through this 4-step process to model coral recruitment that these different recruitment modeling scenarios can be used in either data rich or data poor scenarios. And so I think that in other areas that don't necessarily have all this recruitment data that we were privy to, they can actually use it and apply it to their model to estimate what population dynamics. Thank you for the question.
All right, we have a question from our live audience. Amanda's going to pass the mic over. Just let me know if we can't hear Caroline.
Okay.
Hi, Caroline. That was fantastic. I had a quick question about parietes. So your research showed that it wasn't as thermally tolerant as you hypothesized. And I was kind of wondering if there was any prior research that you came across that had been done on parietes, like documenting if it's thermally tolerant, or is it just because it's such a massive coral that people just kind of hand waved, yeah, it's probably tolerant. Like I was kind of wondering where that came from and how some of these how some of the assumptions in the prior literature got made, if you knew.
Yeah. So I will say from what I've found, pretty much all the research I found was indicating that Porites and these massive growth forms in general are pretty resistant to bleaching. So I'm sure that there are studies out there. I haven't found any that found them to be this susceptible to bleaching. So I think that was one of the probably the most surprising things about my study is that massive Porites was so negatively impacted, because this goes against a lot of other studies, and not just in other areas, but also in Hawaii as well.
And do you think it might be like an exception to the rule, or do you think that maybe that that rule in general might be caused to like put it under a little more scrutiny?
Yeah, I mean, it is possible that again, I don't think I found many other studies that use this type of robust population model for mounding Porites corals, and they're a little bit smaller scale or shorter time periods. So it's possible that just by looking at individual colonies over time, you're not seeing this kind of population level effect that we were able to see with our modeling methods.
All right, thank you.
Thanks.
Yeah, we have another live audience question. Caitlin, go ahead.
Okay. Hi, I really loved your thesis. I was wondering, from the literature that I've read, a lot of focus is placed on like reef building corals, specifically Acropora. So I was just wondering what your thought process was behind choosing these corals. Like, I know that they're supposed to be, some of them are thought to be more stress tolerant, but why these ones specifically?
Yeah. Acropora is definitely a highly researched coral. However, in our sites, they just were not very prevalent. Basically, they're only found in the northwestern Hawaiian Islands. And so, similar to the issue we ran with Thalassopora, we just would not have had the data to do the type of modeling we wanted to do. And the species and actual genera that we chose, these are the three dominant genera in Hawaii. So we looked at 7 species, and I think we pretty much hit 7 of the top 8 dominant species. The only one that we did not include was Porites compressa. It's just like little tiny fingers, and that was partially because it would just be an absolute pain in the butt to annotate. It wouldn't be super accurate. So yeah, they were chosen because those are the dominant species, and then the dominant genera.
Okay, thank you very much.
Welcome.
Okay, we have another,
Sorry.
okay, so we have an online question from Nate Jew, and then we have another audience member question. So go ahead, Nate.
Hey, Nate.
Hey, congratulations, Caroline. Great presentation. I just had a question. I was curious about, it's kind of a modeling question, but when we think about corals, a lot of times people talk about multiple stressors being involved in threatening corals. And you talked a little bit about this at the end. I was wondering if you could speculate a little bit on, like, if you wanted to sort of improve your R-squared, include some other things, maybe think about how you might take your model and apply it to different regions. What are some of the other factors that you think should be included in a model like this to sort of increase its broader applicability specifically? And along those lines, sort of just a minor thing too, I was just kind of curious, were you able to account for how fragmentation of corals might also be contributing to growth and population growth?
Yeah, so I'll answer that one first, so I don't forget it. So we did look at fragmentation and included that as a a random effect in a LME, and found that the model was not had significantly more explanatory power by including the number of fragments. So we were able to luckily drop that from our analysis. We started down the rabbit hole of figuring out how to include that in the model. And I think that would be definitely interesting to look at for the future. But again, for ours, we did find that it didn't improve the model fits. In terms of, so it was what other variables could impact the corals?
Yeah, like how would you, like, what do you think would be you could add to this model to make sure that you're accounting for these effective multiple stressors that
Yep.
might apply to other regions too, right? So
Yep. Yeah. So I think one of the things that would be really great to do was in this whole, like, looking at other environmental variables, one would be just looking at other metrics of thermal stress. So this could be the time since the last disturbance, the high frequency temperature variability, which is basically the daily temperature range. And that variable in general has been shown to really impact corals bleaching impact, as well as coral bleaching hotspots, which are location and magnitude of instantaneous heat stress. So all of these other metrics of heat stress, combining these with severity and frequency, I think would give us a much more robust analysis of thermal stress in particular. And then, and the cool thing is, I know Tom Oliver is over there just like ready to do it. He's already started. We started on looking at some of those variables as well. And it definitely would be interesting to continue looking at those variables. But there's also other environmental variables that could definitely be incorporated. So like one of them that I think would be interesting is looking at nutrients, just because nutrients from runoff, which is very important in Hawaii. They use cesspools for their sewage systems, for example. And nutrients can either positively or negatively impact bleaching. And for the sites in the main Hawaiian Islands in particular, it would definitely be interesting to see if that provided some more explanatory power to the model. But you could go on and on with modeling. That's what Tom Oliver loves.
thanks, Carolyn. Great job.
Yep.
All right, we have another live audience question.
Great job, Caroline. That was a really great talk. I had a question about your methods. So it seems like the annotation of your mosaic imagery is really time consuming, and it seems like that's kind of one of the main limitations that you have. So I was curious, do you know if there's been work done to automate that process using things like machine learning or other tools, or are you kind of limited by, the method as it stands now?
Yep. Yeah, that is the $1 million question, and actual $1 million grants that NOAA Pacific Island Fisheries Science Center is currently working on, is using machine learning neural networks to try and speed up the annotation. And yes, coral biologists that are involved in photogrammetry all over, I think, are itching for this to be perfected to speed up the annotation process. And in particular, NOAA Pacific Island Fisheries Science Center, they have hundreds of these sites throughout the Pacific Islands. And so I know that NOAA in particular is just very, very excited for machine learning to be, again, accurate and be able to be usable. And it's, I guess I can't read the future, but I wouldn't be surprised if it happens in the next couple of years. And then once that happens, once the annotation is actually feasible from a time standpoint, I think the limits with structure from motion are just infinite.
Awesome. Thank you.
You're welcome.
Live audience question.
We have two questions from the Zoom chat of people who might be having microphone problems. Mariana asks, given the results you found that large corals were very important for population maintenance, what implication does this have for coral restoration efforts that usually outplant small fragments?
Yeah, definitely. So I think it's important to note that, coral restoration, and particularly the outplanting of small fragments, is still important, and that, boosting the recruitment is going to add individuals to the population that will, eventually increase the population growth rate. But as we found, recruitment just may not be quite as important as maintaining coral size, coral survival and size. And so I think that, enhancing recruitment is just so much easier, as I mentioned, than trying to enhance survival, and is still an important part of the toolbox. But I also think that this kind of highlights that, addressing climate change at a larger global scale is also is very important. And that's important to know for both local and international management. Thanks, Mariana.
All right. And Marisol asks, on one of your beginning slides, you showed a figure coupled with different numbers of coral taxa you sampled, sometimes in the thousands. Was this a randomization choice or a product of what was available in the field?
Yeah, so that's it's gonna go all the way back through all the animations. Yeah, so that's looking at just those numbers of annotations. And yes, this was basically just a product of what we found in the field. Oh, there we go. What we found in the field where when we started off this project, I was just focusing on Poslopper colonies just to try and get the methods down. Those are also my favorite corals as well. But we quickly realized that in looking across the sites that there were just less Poslopper colonies. And so this was, again, just due to what we found present in the field. And then again, also, Porites, as we did mention, did have a lot of partial mortality. So there were lots of little chunks of Porites and Montipora throughout the archipelago. Thanks, Maddie.
All right, Caroline. Well, once again, wonderful job. I'm going to go ahead and ask everyone who is online and our live audience if the online community could unmute. And we are all going to give Caroline one last big round of applause. So please join me.
Thanks, everybody. Thanks so much for coming out.
Well, thanks again, everyone. We will now be whisking Caroline away with her committee to ask her a few more questions, and then after that, she will be done. So thanks again for joining us today. Bye-bye.
Oh, it just worked.
Recording.